Welcome to this explainer. Okay, today we are exploring a literal paradigm shift in how we understand metabolic health. For decades, we've looked at liver issues through this really narrow lens. But recent research, it's revealed a fascinating biological superhighway inside your body, the gut-liver axis. By the end of this, you're going to see your digestive system and your liver not as two separate things, but as one deeply integrated biological engine. Let's dive right in. To understand why this matters, we kind of have to look at the staggering scale of the problem. Right now, about 30%, yeah, you heard that right, 30% of the entire global adult population is suffering from MASLD. That's metabolic dysfunction-associated steatotic liver disease. And look, this isn't just a harmless bit of extra fat. If we leave it unchecked, MASLD relentlessly progresses to MASH, which brings in severe inflammation and can eventually lead to fibrosis, cirrhosis, or worse. It's skyrocketing right alongside obesity and type 2 diabetes. Okay, let's look at the evolving science here. Notice the stark contrast. The old dogma basically told us fatty liver was this isolated issue, right? Caused purely by obesity, a high-calorie diet, and insulin resistance. Well, it turns out we might have literally been blaming the wrong organ all these years. The cutting-edge view shows us that metabolic liver disease is actually an inside job, driven by that gut-liver axis, meaning a microbiome imbalance in your intestines paired with a leaky gut is actively fueling the destruction of your liver. They are intimately connected. To really hammer home how powerful this connection actually is, look at this dramatic, inciting incident from a landmark study. This was published by Duke University researchers in July of 2026. They found out that merely repairing the intestinal barrier was sufficient to reverse severe metabolic fatty liver disease in experimental models. I mean, think about that for a second. They didn't target the liver fat directly at all. They fixed the gut, and the severe liver disease basically reversed itself. That single discovery is a total game changer for our entire approach to metabolic therapy. So how exactly does this work biologically? Well, here are the 5 critical mechanisms governing this interconnected system. Mechanism 1 is microbiome remodeling, then intestinal barrier integrity, ammonia control, hepatic lipid homeostasis, and finally the urea cycle. All right, diving right into mechanism 1, microbiome remodeling. We're transitioning into the microscopic world of your digestive tract, which is home to trillions of microorganisms. Now, when this gut environment gets out of whack, bad things happen. Pathogenic hyperammoniagenic bacteria, like Clostridium perfringens and certain E. coli strains, they take over. And these specific bugs, they're basically factories pumping out massive amounts of toxic ammonia and highly inflammatory endotoxins, like LPS. That toxic load is what kicks off the liver damage. So the absolute crucial first step in any modern therapy is restoring the beneficial bacteria. Powerhouse strains like Akkermansia mucinifila, Lactobacillus, and Bifidobacterium actively suppress those bad bugs and restore the peace. Moving on to mechanism 2, intestinal barrier integrity. So here we're looking at the actual physical border between your digestive tract and your bloodstream. It's a barrier made of a protective mucus layer, epithelial cells, and these little structures called tight junctions. When this barrier breaks down, which you've probably heard called leaky gut, the biological glue fails, and the gates are just left wide open. Those dangerous ammonia and endotoxins we just talked about, they slip straight through the damaged wall and into the portal vein. It acts like a direct express highway, carrying toxins straight to your liver, where they trigger the liver's immune sentinels, the Kupffer cells. This kicks off a whole cascade of oxidative stress, severe inflammation, and eventually fibrosis. Which brings us to mechanism 3, ammonia control. Now, focusing on the specific chemical threat that's wreaking havoc on the system, you know what's really fascinating about this data? It's the timeline. Historically, doctors thought high ammonia levels, hyperammonemia, were really only an issue in late-stage failing livers. But modern science actually reveals that urease-positive bacteria in your gut start degrading dietary proteins into ammonia super early on. This excess ammonia hits in the early stages of MASLD and is a massive primary driver of liver inflammation and mitochondrial dysfunction. It literally poisons the liver's energy centers. So targeting and reducing that intestinal ammonia production and absorption, that's absolutely crucial for modern therapy. Next up is mechanism 4, hepatic lipid homeostasis. Zooming out of the gut and directly into the liver itself, we've got to look at how fat is actually processed and stored at the cellular level. A healthy liver maintains this beautiful, delicate balance between synthesizing new fats, burning them for energy through oxidation, and clearing out the cellular junk through processes like autophagy and lipophagy. And this whole balancing act is controlled by complex biological pathways like AMPK, PPR-alpha, and SREBP1C.
But when your gut is leaking toxins into the liver, these pathways basically short-circuit. The liver starts hoarding fat instead of burning it. Just trying to reduce inflammation isn't going to cut it. If you don't biologically flip the switch, say activating AMPK to tell the liver to burn fat, that unresolved lipid metabolism is inevitably going to lead to chronic inflammation and fibrosis.
And finally, mechanism 5, the urea cycle.
This right here is the liver's built-in heavy-duty detox system. Under normal, healthy conditions, when ammonia arrives at the liver, it uses urea cycle enzymes to transform that highly toxic ammonia into harmless urea, which your body then safely gets rid of. But here is the really vicious cycle of MASLD. The disease actively dysregulates those exact urea cycle enzymes. So right when a damaged gut is flooding your liver with totally unprecedented levels of ammonia, the liver's own machinery to clear that ammonia breaks down. The liver literally becomes overwhelmed by the exact chemical that's attacking it from the gut.
Now let's look at targeted ingredient mapping using hepatoferment 3 xbiotics. So how do we practically apply all this incredible science? Well, let's look at an objective case study of a formula called hepatoferment 3 xbiotics to see how researchers intervene across the entire gut-liver axis, and this brilliantly illustrates the approach. For mechanism 1, microbiome remodeling, specific probiotics like Lactoplantobacillus plantarum are mapped because they actively control that nasty ammonia-producing C. perfringens we mentioned earlier. Plus, fermented cranberry and pomegranate support the beneficial Akkermansia. Then, for mechanism 2, the intestinal barrier, ingredients like col kefir are mapped to give primary structural support to the intestinal mucus layer, alongside fermented Porreococos and astragalus to really tighten up those leaky cellular junctions. It's an incredibly precise approach. Moving right along to mechanism 3, ammonia control, we see that the formula doesn't just chemically bind ammonia. It actually uses strains like B. lactis to shift the gut environment, which reduces the proteolytic bacteria creating the ammonia in the first place. Pretty smart, right? Then for mechanism 4, lipid homeostasis, potent fermented botanicals are deployed. Fermented turmeric and Scutellaria bicalensis are mapped specifically for their ability to activate that AMPK pathway and promote autophagy. Meanwhile, fermented cordyceps targets mitochondrial beta oxidation to help the liver actually clear out that accumulated fat and restore energy balance. Finally, for mechanism 5, researchers mapped a really specific compound, L-ornithine L-aspartate, or LOLA for short. The inclusion of LOLA here is fascinating because it specifically addresses the liver's overwhelmed detox machinery. It directly targets the urea cycle, rescuing the liver's ability to transform that highly toxic systemic ammonia into harmless urea while supporting glutamine formation. It's absolutely essential for hepatic nitrogen detoxification, basically giving the liver the exact tools it needs to clear the backlog of toxins while the gut is busy being repaired. So what does this all mean? Well, this case study shows us that the paradigm has officially shifted. Treating fatty liver disease is no longer just about telling someone to lose weight and crossing our fingers that the liver fat melts away. No way. The future of MASLD treatment requires a holistic, simultaneous approach. We've got to remodel the microbiome, seal up that leaky gut barrier, control ammonia production, jumpstart the liver's lipid metabolism pathways, and optimize the urea detox cycle, all at the exact same time. It really makes you look at your body like a giant puzzle, doesn't it? The science is incredibly clear now. These systems are just not isolated. So I'll leave you with this final thought to ponder. When you think about protecting your liver, managing your weight, or optimizing your energy, does your metabolic health actually start in your gut? I highly encourage you to dive deeper into this fascinating topic. Thanks so much for learning with me today, and keep exploring.